All right, so in our first lecture, we talked about the light-dependent reactions, which were the portion of photosynthesis that required sunlight directly. So this was sort of a summary of what we talked about in our last lecture. So in the thylakoid membranes of the chloroplast, which are these, we have these photosystems. And there are two different ones, photosystem 2, photosystem 1. We didn't get into a lot of the details of the differences between them, but they work together, really, in order to make the two products that are necessary for the Calvin cycle, which is what we're going to talk about today. So when light hits a photosystem, it excites the electrons. The electrons travel down an electron transport chain. You can see that happening here and here. And those electrons power hydrogen ion pumps. So these hydrogen ions are also called protons. And these protons are going to build up here inside the lumen, which is the inside of these thylakoid sacs. So inside of these is where the hydrogens are building up. And that creates a concentration gradient. The hydrogens really want to get back out into the stroma, and their only pathway is going to be through ATP synthase. And as they flow through ATP synthase, that is going to take the ADP and the phosphate, hook them together, and make ATP. So our first product of the light reactions was ATP. It's captured the energy from the sunlight, and it's going to carry that energy in the form of chemical energy and use it to build sugar. The second product, one of these electrons will also join up with some of these hydrogen ions, these protons, and it'll turn NADP plus into NADPH. That's our second product, NADPH, and that's important because it's going to bring the hydrogens to make glucose. Because remember, glucose is C6H12O6. The hydrogens are getting carried over there from NADPH. Now, an electron left, originally here. To replace the electron that left, water was going to be split. That water is going to actually break into three things. It's going to break into proton, which adds to this proton gradient. It's going to break into an electron, that's the missing electron. That's going to replace the electron that left when it got excited from the light hitting. And it's going to break into oxygen. And the oxygen, when two of those get together, that's going to become the oxygen gas that's released from the plant, and that's the oxygen that creates for our whole planet, the oxygen for our planet. It's going to come from plants and algae releasing the oxygen from the splitting of water. Okay, so the light reactions in water and sunlight, also NADP plus and ADP, which are going to get turned into NADPH and ATP. The oxygen from the water is going to get released into our atmosphere. The hydrogens from the water, that's going to become get carried over to the Calvin cycle by the NADP. Okay, so these products are going to become part of glucose later. The hydrogen here, and then the ATP, the energy, is going to be used to build the glucose. Okay, and so that brings us to the Calvin cycle. So the Calvin cycle, or light-independent reaction, this is going to be where carbon fixation actually happens. In other words, this is where the carbon dioxide is going to get converted into glucose, which is called fixing carbon, or carbon fixation. So this is a metabolic pathway. We learned about those in the last chapter. Enzymes are involved at each of these steps. Enzymes, 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 enzymes. And it's kind of a cycle. Carbon dioxide comes in. It's going to, the carbon from the carbon dioxide is going to take this 5-carbon molecule and turn it into a 6-carbon molecule. That's going to split into 2 3-carbon molecules, and it's just going to go through a whole bunch of steps, and you actually do not have to know all of the details of the individual steps here. What I want you to notice here, really, in this overview, a couple of things. One, it takes 6 carbon dioxides to build a glucose, because remember, glucose is C6. So we need 6 carbon dioxides. That's going to be our 6 carbons. So this is actually going to turn 6 times. It's going to take those carbon dioxides one at a time, and it's going to go through several steps, and then take a second one and go through several steps, and ultimately, after 6 turns, we're going to get 1 glucose. That's the first thing that's important. The second thing is, notice what's required for this, ATP and NADPH. Again, the hydrogens from the NADPH, notice NADPH is just turning back into NADP+. the hydrogens are getting added in. This is going to become the glucose, the hydrogens of glucose. And the ATP are providing the energy for some of these steps that are endergonic. So this is kind of an overview. It's a step-by-step process. Now, we can break it down into kind of 3 main stages. Again, you do not have to know the major details of these stages. So the first one is that the carbon dioxide comes in. Again, it's going to hook with this 5-carbon molecule here called RUBP. You don't have to memorize RUBP. But it is kind of important that you know this first enzyme, rubisco. Notice this rubisco is like the nickname for the enzyme for this step. This enzyme is what's going to hook this carbon dioxide in. It's going to turn into a 6-carbon compound, which is very unstable, and that splits into 2 3-carbon compounds. So it's going to go from 5 to 6 to 3, and the carbon from carbon dioxide is coming in. And this is called PGA. So that's our first step. The next step is where the NADPH and the ATP are going to come in. These are going to be made to build something called G3P. Now, to make a single G3P, glyceraldehyde 3-phosphate, that leaves the cycle, it takes 3 turns. So 3 turns is going to have 1 G3P leave this cycle. That's a 3-carbon molecule. And then what's going to happen is, in the next step, the ATP is going to be used to basically turn this back into its original form so that it can take in the next carbon dioxide. So it's 3 main steps. You do not have to know the details of these for AP exam purposes. What you really need to know is, we're going to talk about this enzyme rubisco in a second. That's important. And know that basically 3 turns of this makes 1 G3P. And then what ends up happening is 2 of these G3Ps hook together to make glucose. So it doesn't actually make glucose like 1, 2, 3, then 4, 5, 6, like adding 1 carbon at a time. It makes a G3P, then it makes a second G3P, and then those hook together, and that becomes the glucose. The numbers of ATP, that's kind of beyond the scope of this course. How many ATP do you need? How many NADPH do you need? Not overly important. Just know it's a cycle and kind of the number of turns, and that ATP and NADPH, kind of the general role they play. Okay, so photorespiration. So here's the reason that I mentioned rubisco. It's ribulose 1,5-biphosphate carboxylase, which is a really long name, so we just call it rubisco. And here's the thing. That enzyme is attracted not just to carbon dioxide, which is what we want it to go to. If it joins with carbon dioxide, it turns carbon dioxide, if we go back for a second, it turns carbon dioxide, let me erase all this work here. Okay, so here's rubisco. It takes carbon dioxide, it makes this 6-carbon molecule that breaks into the 3-phosphoglycerate. That's what it's supposed to do. But it's also attracted to oxygen. So remember competitive inhibition. So what happens is, if it binds to oxygen, it ends up just making more carbon dioxide. So basically everything that happened in the light-dependent reactions just goes to waste. The plant just releases carbon dioxide back out and makes no sugar. I mean, ultimately a plant that was doing high levels of photorespiration, where it was using oxygen instead of carbon dioxide, it would die because it would not be able to make enough sugar to stay alive. And so oxygen is almost like a competitive inhibitor. It's getting in the way. So the more oxygen there is in the environment, the more likely the plant's not going to be very efficient at photosynthesis, because rubisco is going to bind to the wrong thing and waste all the work that was done in the light reactions. And this is kind of a nice little diagram of it. So rubisco is attracted to both oxygen and carbon dioxide. It's supposed to bond to carbon dioxide, in which case then it goes through the whole thing and it ultimately makes sugar. But if it binds to oxygen instead, it wastes all the ATP, the energy of ATP, all of that gets wasted, and the oxygen binds to carbon and makes more carbon dioxide. And so the plant ends up not making sugar. So here is the solution to the problem. It is called C4 photosynthesis. Now the reason it's called that is because the original Calvin cycle, if you remember, if we go back for a second, if we go back a couple slides here, the original photosynthesis, the first thing that it made was a 3-carbon molecule, right? It took this, made the 6, it split into 2 3s. So sometimes photosynthesis that just uses the Calvin cycle directly, just regular photosynthesis, is sometimes called C3 photosynthesis. So with C4 photosynthesis, the modifications are, first of all, it has a special enzyme called PEP carboxylase. So in the carbon dioxide, this is supposed to be the stomata where the carbon dioxide comes in the leaf. Normally, carbon dioxide and oxygen can both enter here, and in a high oxygen environment, this is where the photorespiration would happen. In a C4 plant, the plant is modified so that the Calvin cycle happens in an area further inside the leaf. And instead of the carbon dioxide going straight to the Calvin cycle, it bonds to this enzyme called PEP carboxylase instead, making a 4-carbon compound called PEP. And that 4-carbon compound gets transported further in the leaf, and then the carbon dioxide is dropped off. So it'd be sort of like if you open your front door and you let somebody in, but you didn't want to be in contact with any people, so you're behind a second door. And you have a messenger that opens the front door, grabs the package, and then takes it to you further inside. So this would be the front door. PEP carboxylase is not attracted to oxygen. It picks up the carbon dioxide, carries it, and dumps it off further inside. No oxygen can get further into this area. And now the Calvin cycle can happen without oxygen getting in the way. And so that is called C4 photosynthesis. So the leaves have a special structure, and it uses this special enzyme to carry it further in. Notice it still does do the Calvin cycle, though. This doesn't replace the Calvin cycle. Okay, and then there's another thing that can happen. So plants that live in the desert, they have another problem, which is water loss. Whenever the stomata open, water evaporates out, just the same way that when you exhale, you breathe out water vapor, right? So every time you breathe, you're losing water. You can't really help it. Well, plants lose water when they open their stomata, which are the pores underneath the leaf. At least they're underneath, which helps. But when they open those to get the carbon dioxide for photosynthesis, some water escapes. So plants that live in the desert, they can't afford to lose water. So what they do, interestingly, is something called CAM, or crassylation acid metabolism. So normally, the carbon dioxide is collected during the day, because remember I told you that you can't store the ATP, you can't do photosynthesis at night. So normally, the light reactions in the Calvin cycle are happening at the same time. In CAM, they're still happening at the same time, but the plant collects all its CO2 at night when it's cooler, and that way it's not going to lose a lot of water. It stores it as an acid called malic acid, and then during the daytime, it still does the Calvin cycle during the day, like regular photosynthesis, but it doesn't have to open its stomata. So imagine if you could do all your breathing at night and then just hold your breath all day. You didn't have to open your mouth during the day. That's basically what they're doing. They still only do photosynthesis in the daytime, but because they can collect their carbon dioxide at night and store it as an acid, then they don't have to open their stomata to get their CO2 during the day. They can leave them closed and then just do photosynthesis during the day without opening the stomata and without losing a lot of water. And that's all this says. So water is lost. That was called, we learned about this before, it's called transpiration. And so to prevent the water loss, the CAM plants open the stomata at night. They do not do photosynthesis at night. They just store the carbon dioxide until the daytime. And the name comes from the fact that it was first discovered in plants in the family Crassulaceae. I can't even pronounce it. Crassulaceae, which look kind of like this. It's also done in cactuses, but that's where the name crassylation acid metabolism. The acid is not called crassylation acid. It's actually malic acid is the acid that's made, but we call it CAM for short. So these are two modifications, C4 and CAM, that can allow photosynthesis to occur in either high oxygen environments, which a lot of plants use C4. You could look up which plants use it. And then desert environments for CAM.